Okay, today we will be discussing the spleen, and it's a lymphoid organ, and I will take two spleen viscera in the lab. You please see. I will explain to you, and then if you know the gross anatomy, you will be correlating with histology. Now, competency-based undergraduate curriculum. Now, they identify the lymphoid tissue under the microscope and describe microanatomy of lymph nodes, spleen, thymus, tonsils, and correlate the structure with function. Now, today we will be discussing the spleen. Okay, it's a lymphoid tissue. It's a lymphoid tissue. Now, lymphatic system provides protection to individuals by imparting immunity. Immunity is the ability of the organism to resist a particular infection or toxic substance with the help of antibodies or activated white blood cells. There are two types of immunity, innate and acquired immunity. Innate or native immunity is present at birth. Innate immunity provides non-specific protection against all types of infections. Acquired or adaptive immunity is acquired by an individual throughout life. Now, acquired immunity is of two types. This is a viva question. The active immunity, it is acquired by an individual on exposure to antigenic agents, toxins or microorganisms. During exposure, immune cells get activated and impart acquired immunity. Passive immunity, it is acquired by an individual by the transfer of ready-made antibodies, immunoglobulin injections. Okay. Passive immunity, it is acquired by an individual by the transfer of ready-made antibodies. Ready-made antibodies by immunoglobulin injections. Immunoglobulin injections. Now, immunity protection by immune cells or antibodies, innate immunity present at birth, non-specific protection. Acquired immunity, active immunity naturally acquired by infection, artificially acquired by vaccination. Now, passive immunity acquired by transmission of antibodies, for example, transfer of maternal antibodies to fetus, immunoglobulin injections. Now, lymphatic tissue, list the primary and secondary lymphoid organs. List the primary and secondary lymphatic organs. Lymphatic tissue is a specialized form of connective tissue that consists of immune cells, lymphocytes and plasma cells, meshwork of reticular fibers and cells. Lymphoid tissue identifies foreign antigens and dead or old cells of the body and removes them by phagocytosis or by producing antibodies. Lymphoid organs are classified as primary and secondary lymphoid organs. Now, primary lymphoid organs, these are called central lymphoid organs. These organs produce immature lymphocytes from stem cells or immature cells. For example, thymus and bone marrow. This is a question for NEET. Now, these are also called central lymphoid organs. These organs produce immature lymphocytes from stem cells or immature cells. For example, thymus and bone marrow. Now, lymphoid organs, primary lymphoid organs convert stem cells into immunocompetent cells. Example, thymus, bone marrow. Primary lymphoid organs convert stem cells into immunocompetent cells. Thymus, bone marrow. Example. Now, secondary lymphoid organs site for activation and proliferation of immunocompetent cells. Example, spleen, lymph nodes, tonsils, appendix, and secondary lymphoid organs. These organs provide exposure to foreign antigens and convert immature lymphocytes into active lymphocytes. Now, secondary lymphoid organs, these organs provide exposure to foreign antigens and convert immature lymphocytes into active lymphocytes. For example, spleen, lymph nodes, tonsils, appendix, and Peyer's patches. Histologically, lymphoid tissues are grouped as follows. diffuse lymphoid tissue and dense lymphoid tissue. Now, diffuse lymphoid tissue, lymphocytes are distributed throughout the mucosa of gastrointestinal tract, respiratory, urinary and reproductive tracts. This type of lymphoid tissue is called diffuse lymphoid tissue. As this lymphoid tissue is associated with mucosa, it is also called mucosa associated lymphoid tissue, MALT. Now, as this lymphoid tissue is associated with mucosa, it is also called mucosa associated lymphoid tissue, MALT. Ileum is the most common site for MALT. This can be an MCQ question, it can be a NEET question, it can be a short note on diffuse lymphoid tissue. Now, it protects and provides immunity against microorganisms coming in contact with mucosa. Diffuse lymphoid tissue is not enclosed within the capsule. Identification features. Now, diffuse lymphoid tissue is not enclosed within the capsule. Identification feature. Some authors consider MALT as a dense lymphoid tissue. Now, dense lymphoid tissue is enclosed by well-defined capsule. Identification feature, it includes discrete lymphoid organs such as lymph nodes, spleen and thymus and tonsils. Now, a well-defined capsule, when you see the now it can be this. Now, the splenic artery, splenic vein and splenic artery. Okay. Now, if you this includes a capsule. Now, cells of the lymphoid system. List the cells of lymphoid tissue. Lymphoid tissue consists of lymphocytes and other supporting cells such as reticular cells, epithelio-reticular cells. Some cells of immune system also forms part of the lymphoid tissue. They include monocytes, macrophages, neutrophils, basophils, dendritic cells and so on. Differentiation cells of lymphoid tissue depends on the presence of specific molecules on their surface. These molecules are called cluster of differentiation molecules or markers. Now, lymphocytes is a subtype of white blood cell that forms a major component of lymphoid tissue. White blood cell. Lymphocyte is a subtype of white blood cells that forms a major component of lymphoid tissue. Now, these are the cells of the lymphoid tissue. So, they have a central round nucleus and thin rim of cytoplasm. Nucleus is densely stained, heterochromatic. Identification features. Size small lymphocytes 7 to 10 nanomicrons and large lymphocytes 4 to 20. Histologically, lymphocytes cannot be differentiated into subtypes. This histologically, you cannot differentiate the lymphocytes. You cannot differentiate into subtypes. Based on the presence of specific CD molecules, lymphocytes are grouped into three types: T lymphocytes, beta lymphocytes, and natural killer NK cells, MCQ. Now, based on the presence of CD molecules, lymphocytes are grouped into three types: T lymphocytes, B lymphocytes, and natural killer NK cells. Natural killer NK cells, natural killer NK cells, MCQ. All lymphocytes originate from bone marrow. You have to know where does it originate from. Now that is from the bone marrow. Okay, T lymphocytes also originate from bone marrow. Now undifferentiated lymphocytes originate from bone marrow and enter the thymus. In thymus, they get differentiated into mature T lymphocytes. They maintain cell-mediated immunity, self-mediated immunity, VIVA. T cells have a specific T cell receptor, TCR. TCR recognizes antigens bound to the cell membrane and proteins called major histocompatibility complex, MHC molecules. Exposure of T cells to specific MHC molecules convert T cells into effector or memory T cells. Memory T cells maintains population of T cells that get easily activated in reinfection by the same microorganism. These cells persist for a long time. Ekari thaglo, come see the viscera, see the spleen and see the slides. And see, you can see now it is related with the diaphragm. The lateral surface is related with the diaphragm. So inspiration, expiration, that spleen moves. You can, if it is now this is the spleen. Now these splenic vessels, they go to the sinus, similarly as the kidney. Okay, now this is in the diaphragmatic, related to the diaphragm. Medially to the abdomen, the tail of the pancreas comes and sits here. Now the splenic artery and vessels are going there. Now that ramifies, that distributes into the district. You will be seeing whatever you are seeing. You will see that this is lined by the capsule, then the white pulp, red pulp, and the capsule, it intervenes between the thing. Now this is more or less a natural, a normal spleen, but you can get this big spleen, this big. एकबारे बड़ो, that means for immunity and these things, लड़ाई करने के लिए बढ़ जाते थे. Now this, with this idea, you open, you see the slide, see the atlas. If you have any problem, I'll be showing it to you. But you feel, you see. Today you might not be coming, you have not brought your gloves, but when you see in your thing, viscera, hath diye dekhe. You have to feel it. Until unless you feel, touch, you cannot become a doctor. So AI, chat GPT and all this robotics and some painting, AI, GPT painting is going on. It's a craze now throughout the world. Okay. Anyway, let's talk business. You can see it in the, I put it in the center. Now, see, when you get large kidneys, you even get large kidneys. This is kidney, this is a spleen. By seeing it has a hilum, it has a hilum. Now, this is the ureter, these are the renal artery, renal vein. Now, when it is lying like this, you are puzzled. MS exams, 48 hours. 48 hours in a day, 24 hours in a day. The exams sara din raat jege achi. So at least you will be having a panic syndrome. Definitely everyone has. Now you have to see there might be, this is small kidney. Okay, it can be an average kidney of this size. You will see it over there. Many bodies are there, but this is see, it is having a capsule. So you just, I want to make it this thing, that this is the spleen, this is the kidney. Now, in histologically, whatever it is, the processing, the sections you do, it might be longitudinal section, it might be a transverse section. So, the vessels and the the collecting duct, collecting tubules, according to the section, if it is a transverse section, then it will come like this. Oh, if it is a LS section, then it will come with the features, with the calyces open and everything.